Okay, so the picture that you have is of a mitochondria. We're looking at aerobic respiration. It's aerobic because it has oxygen. You want to do aerobic respiration, not anaerobic respiration if you have a choice, because anaerobic respiration makes how many ATPs?
0.
No, not 0. 2. Aerobic respiration can make a max of 36. 36, that's okay. Huge difference, right? Huge difference. That's easy. Look, if you could do a job, you can do the same job, but you can get paid $2 an hour, you can get paid $36 an hour. I mean, we're going to have to send you back home to mom if you don't choose the 36, right? That's an all-day no-brainer. You might as well get paid. It's the same idea here. Okay. True or false, glucose is the only biomolecule that can be used for respiration.
No. False.
False. True or false, you can use lipids, fats, oils, waxes. True or false? True. True or false, you can use protein.
True.
True. True or false, you could use DNA. True. True or false, you could use other sugars.
True.
True. Yeah, you can use almost anything. Okay. People who want to lose weight, they go on a diet. They diet too severely. Well, first of all, when you do go on a diet, like I need to get back on. Well, I'm doing okay, but there's some changes I need to make again. Why do you don't have to be polite? Why does Andrew want to go on a diet? Don't be polite. I'm asking you to be real. Which part of me, do I want to lose these? No, I want to lose this, right? I want to lose fat, which should tell you right away that what I'm trying to do is get my body to, instead of using sugar for energy, to use fat for energy. Which should tell you that your body can use fat for energy, right? It can use lipids for energy. Here's the problem. People go crazy with their diets, they do it wrong, they try to go too fast, they shut everything down, and instead of burning just the fat or burning exclusively the fat, their body starts to burn the muscle, the protein, and they lose muscle mass. Just something to think about. There are different ways to diet well and healthy and all those kinds of thoughts. So I say all that to say, your body doesn't just use sugar. Now your body will preferentially use sugar, okay? If you eat a steak and drink a milkshake, your body's going to use the milkshake because it's easier. If you drink a shake and a cup of oil, your body's going to use the milkshake preferentially because it's easier and it's just going to store that oil. Well, that's dumb. Why would you want to do that, right? For that matter, if you're trying to diet, don't drink the milkshake. Skip the milkshake. Eat protein. Eat lots and lots of protein. All right. So you can use multiple biomolecules. You can eat almost anything and use it for energy. Your body really likes glucose, though. It uses it all the time. If you're low on energy, your body tells its cells to release glucose that it's stored, to release it back out because it's a ready supply of energy for the body. Okay? So that's the reason that we use glucose as our model. Let's think about respiration for a second. If we're doing glucose, do you know the formula for glucose? You must. If you don't already, you must know it. What is it?
C6H12O6.
C6H12O6. Good. Yeah. C6H12O6, right? Hydrocarbon, if you were to break this down, for every carbon there's one hydro. CH2O is the generic formula. It sounds like ose. Remember, we've been talking about this since like the second week of school, right? So carbohydrate, C6H12O6, this is glucose. We have a formula for the complete respiration of glucose. C6H12O6 plus, what do I need to burn my glucose in aerobic respiration?
Hydrogen. No.
Think about just the name of it, respiration.
Oxygen.
You need oxygen. How many of these do I need? No, sir.
6?
You need 6. Okay? Now, folks, if I am coming back to this like the 4th time, you got to tell me this. This will be on a test. You can count on that. C6H12O6 plus 6O2 yields. What do you make out of respiration? What do you breathe out?
Carbon dioxide.
Carbon dioxide, CO2. How many of those am I going to make? You just had to take a guess. 6. How many carbons do I have here?
6.
6. How many carbons do I have here? 6. This is my only carbon source for output, right? So if I had 6 carbons here, I got to have 6 carbons here. Okay? Now, the other byproduct of this is
Water.
water. How many? It's very convenient.
2.
6. Okay? This balances. I got 6 oxygens plus 12 oxygens is 18 oxygens. I got 12 oxygens plus 6 oxygens is 18 oxygens. I got 12 hydrogens. I got 12 hydrogens. 6 carbons, 6 carbons. This is a balanced formula for aerobic respiration, right? This would look differently in anaerobic respiration. All right. So, we got glucose. If you look at your deal here, it's being broken down in glycolysis. What does glycolysis mean? Glyco means sugar. Lysis means splitting. So glycolysis, glycolysis means splitting of sugar. We take that 6-carbon glucose and we split it in half, right? Into 2 3-carbon molecules called pyruvates. You're going to have to know that name. Okay? In a minute, this is not your review. Well, this is a review sheet, but I'm going to hand you a couple things, just so you know. You're going to get hooked up today. I'm going to give you one of these that's blank. Actually, I'm going to give you 2 of these that are blank to practice with. Okay? And I'm going to give you this beautiful thing with 32 questions. All right. So we immediately split the glucose into 2 things, right? What's produced? 2 things are produced during glycolysis.
Pyruvates.
Well, I'm sorry, 3 things. We got pyruvates. What else?
ATP.
ATP. How many?
2.
2. Good. And the other thing we make is? Go ahead and look. It's on there.
NADH.
NADH. What does NADH have?
Hydrogen.
No. Well, yes, it does.
Electrons.
Electrons. It's an electron carrier, right? To split the glucose into pyruvates, we have to take electrons away, right? Those electrons have a lot of potential energy in them. A lot. Remember, we only make 2 ATP in glycolysis, but if we go to the Krebs cycle and to the electron transport chain, we can make up to 34 ATP. It's huge. Okay. This first step, look at where it's happening in the cell. Where's it happening? Is it in the mitochondria?
No, outside the cell.
Well, that's a mitochondria. We're inside the cell with all of this, but we're outside the mitochondria. It means it's in the cytoplasm. Glycolysis happens in the cytoplasm. You can see it, right? I mean, right now. Okay. Then these molecules have to move into the mitochondria, the NADH and the pyruvates, which is where the next couple steps happen. The NADH that it just made can go straight to the electron transport chain. The pyruvates, they go to what we are calling the link step. It's where we take 2 pyruvates and we convert them into, what molecules that say there?
NADH.
Take a look at it. Well, okay, it does produce some NADH, but that's just carrying electrons.
NAD.
But notice, take a look here. You've got 2 3-carbon molecules, and it gets converted into 4 byproducts. Kind of looks like this. We went off here and we said, hey, there's 2 of these. And we went over here and we made these. These all have carbon in them, right? Well, these things are CO2. These things are called acetyl coenzyme A. Okay, so these are pyruvates, right? They came from my glucose, it got split, made 2 pyruvates inside the mitochondria during the link step. This pyruvate is converted into acetyl coenzyme A, which is a 2-carbon molecule, and 2 CO2s. These carry on to the Krebs cycle, where they're ultimately converted into 4 CO2s. When I split these molecules, when I'm converting the glucose into this, remember I take electrons out. When I convert this into these 2 things, I take electrons out. When I convert this into this, I take electrons out. I keep taking electrons out. Every time I'm splitting a molecule, I'm stealing its electrons, right? There's energy in those electrons. Those electrons are going to power the electron transport chain. So we're going to keep making NADH. There's also a molecule called FADH2 that's made at the end of the Krebs cycle. I'm a little less concerned about that, okay? You know the name of that, but you really need to know FADH, okay? It is the primary electron carrier during this. We're storing electrons in there, okay? All right, so the step that's happening outside of the mitochondria is called glycolysis. The first step inside on the far left is called, we're calling it the link step, right? We convert 2 pyruvates into 4 molecules, 2 of each thing. What are they? Acetyl coenzyme A and CO2. The acetyl coenzyme A go into the Krebs cycle, hence the circle wheelie thing that's being drawn there. In the Krebs cycle, the 2 acetyl coenzyme A's get converted into? No, sir. Just follow the carbons. What do the carbons become? Take a look at the arrow coming out of that cycle.
4 CO2?
4 CO2s, okay? It also produces NADHs, produces FADH2s, it produces a little bit of ATP, okay? But to this point, I've still got a whole lot of untapped energy. Where is it stored? Yes, yes, very good. Bonds in which molecule?
Maybe like NAD.
Yes, in NADH, right? It's carrying these high energy electrons. They're going to be very, very useful. Where do they carry them to? Just think of the word electron. What do we have that has the word electron in it that's going to make a lot of energy? Electron transport chain, okay? So NADH and FADH2 are both going to carry electrons to the electron transport chain. Notice where they come in at. NADH comes in at the start of the electron transport chain. FADH2 comes in at the second step of the electron transport chain. They have, the electrons from them have slightly different energies, and so they are, NADH electrons are more efficient for the electron transport chain. One thing I've not drawn on here, but you can, it would be helpful. If I were to draw this, you've got get this big thing, get this little thing, get this little thing, get this big thing, get this thing, And you got this thing. Then I guess you got one more, right? You got ATP synthase, which is kind of a big deal. Okay. So what happens is the electrons from NADH come here. It's carrying 2 electrons. Okay, those electrons go to this first molecule, which is a pump. And the electrons transfer ultimately to this molecule, and then to this molecule, and then to this molecule. Well, as they go, the energy that's in that electron is used to pump H plus ions. I'm just going to draw pluses, okay? It's used to pump them through. Now, where do I keep getting all these H plus ions? Well, for one thing, every time I have an NADH and I take its electrons away, I regenerate the molecule NAD plus, which is, I need this to do glycolysis. If I ever run out of NAD plus, I can't do glycolysis. The whole system shuts down. So one of the major reasons we see for the electron transport chain is that we have somewhere to stick electrons so that we can begin again with glycolysis. I leave my H pluses behind here, and they're actually used, right? The electron moves through, and its energy is used to pump H pluses up here, okay? This is where FADH2 comes in. It's not used to pump anything here because it came in a step later. Its electron will also go over here, and at this stage we pump more more H plus ions through. Okay? Then those electrons continue on to here, and ultimately to here. Okay? They have to have an end. There's got to be an end to these electrons. Okay? They've got to go somewhere. They came from glucose. They were transferred for a while to NADH and FADH2, but where will they end up? What's the final receptor molecule in the electron transport chain? Do you know the answer? It's oxygen. So what happens here is, at this stage, we take O2 plus 2 of these pluses plus 2 of these electrons from the electron transport chain.
Where did we grab
And
the
we-
oxygen from?
Huh?
Where do we get the oxygen from? Oh, okay, this is already there.
Actually, use half an oxygen here. This would get doubled for a full oxygen. You know, because oxygen that you breathe is O2, it's not just O, right? So this would get doubled for,
But like, you're getting the oxygen is just coming back into play because you haven't been using the oxygen, you're transporting all of the.
I haven't used any oxygen yet, right? Yeah. So when you look at this starts in the Krebs cycle, right? This we've been making all along, right? But these don't happen until the last step. You breathe in oxygen, and you breathe out carbon dioxide, and you produce water. So that's what we're going to get here. When I take half an oxygen molecule and 2 protons and 2 electrons, I make water. Okay. That makes sense? This is why it's called aerobic respiration. I've got to have this oxygen in order to do this process. No oxygen, this should. If I don't have oxygen to receive this whole thing backs up. Electrons cannot flow, which means NADH backs up, and FADH2 backs up, which means glycolysis backs up. Because I don't have, remember, I need FAD. Take a look at your thing here. I guess I didn't write NAD plus. You need NAD plus to make NADH. If you don't have it, you can't do it. So you can't do glycolysis. And so you don't make any energy. You could have all the glucose in the world, but if you don't have any NAD+, you can't do a thing with it. Right? Make sense? I don't know, what's that like? If you had everything you needed to make a bunch of ham and cheese sandwiches, but you didn't have a knife, like cut open the package of meat or the mayonnaise, or like, it does you no good. You've got to have something. You can have all the meat and all the cheese, but if you don't have any bread, you don't have a sandwich, right? You've got to have NAD+ to be able to do this. If I don't have oxygen, this backs up. Your body finds another way. Instead of doing aerobic respiration, it does anaerobic respiration. And so then you're going to take your NAD+ with your electrons, you're going to take your pyruvates with your carbons, and you're going to make either lactic acid if you're human, or you're going to make alcohol if you're something like yeast. And that gives you a place to, instead of storing them in oxygen and making water, you store them making alcohol or making lactic acid. And so then you can continue glycolysis. All right. This is not easy. Agreed? Nothing about this is easy. And I've made this as easy as I can. I'm telling you right now, for a test, you've got to be able to fill this out. Okay? The only way I know to do that is to practice, 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 practice. Okay? So you have one of these filled out, and now you have two of these not filled out, and even then, I would probably take a picture of this before I wrote on it, okay? So that you could, and I would just practice, practice, practice. There's nothing for it but to do it.
Are you on a test? Will this be how it's given to us? Like with
Oh, yeah.
Like with all the shapes around it?
Yeah.
I mean, take two for Kevin.
You see?
We got
I
practice.
thought you didn't talk to him like that.
We have practice. Not seeing.
I heard he wasn't that close. I have not written your test yet, but I will. And I'm going to use these questions. It's probably going to be multiple choice. So it probably will not be worded exactly how we're going to word it right now, but it's going to be real close. Along with a bunch of other tantalizing, not correct answers, right? So number one, what is respiration? What is all this stuff that we're talking about, that we're doing? Yeah, it's releasing energy from molecules. Releasing energy from molecules. Or maybe we could say it this way, it's making, what are we making out of this? What's our energy form? ATP. It's making ATP from the energy of biomolecules. Number two, sorry, you're still writing on number one. Making energy, or making ATP from biomolecules, or converting energy from biomolecules into ATP, something along those lines. Okay. Number two, what is the major energy currency of all cells? ATP. You know what other choice I will put on there? It's going to be, you better know glucose is on there, right? Don't pick glucose. Don't do it. Major energy currency of all cells is ATP. You can't do anything with glucose. You can do everything with ATP. Number three, what is the generic formula for aerobic respiration of glucose? Plus 6O2 yields 6 CO2 plus 6 H2O. When in doubt, put a 6. Had to drop it every now and again. That's been weird. Number 4, what is the difference between aerobic and anaerobic respiration? Do not say AN.
One uses oxygen and one doesn't.
Okay, that's part of it, right? One uses oxygen, one does not. It's very true.
One produces more energy and
One produces 2 ATP and the other one can produce up to 38 ATP. That's good. You're good. The first thing is like literally one uses oxygen, one does not. Right, and the other one is that one yields only 2 ATP and the other one yields up to 38 ATP.
38, 36.
It's a total of 38 because you get the 2 from glycolysis and you get another 2 out of the Krebs cycle and you get potentially another 34 out of the electron transport chain. In 5 years, you are not going to remember that glycolysis makes 2 and the other one, and the electron transport chain makes 34, and the Krebs cycle. In fact, you won't remember that in 6 months, probably, right? But can you hopefully remember that like aerobic respiration makes a whole lot of energy and anaerobic makes like almost none? I mean, if we need to have those just large, big picture concepts now. All right. Which types of organisms do respiration? All. If you're alive, you need energy, so everybody does respiration. Number 6, which organisms do aerobic respiration? Which organisms? Yeast can do it. They can also do anaerobic. What else can do aerobic respiration? Which ones use oxygen? Plants, animals, fungus. What do those all have in common? They are what kind of cells? Eukaryotes. Good. Eukaryotes. Eukaryotes do aerobic respiration. I want you to think back about that for a minute. What organelle do you have to do aerobic respiration? Mitochondria. Which critters have mitochondria? Eukaryotic cells. Which ones do not? Prokaryotes. Therefore, they cannot do aerobic respiration. They can only do anaerobic, right? All right, next question. Which organisms do anaerobic respiration? Prokaryotes do, but also eukaryotes if they don't have oxygen, right? So everybody does it. Number 8, which types of organisms do lactic acid fermentation? Why don't you put down us? I don't care if you know that other animals do it. I care that you know that you do it. This is, where do you feel your lactic acid? Your soreness in your muscles, right? When your muscles didn't have enough oxygen, they build up lactic acid. Why did they build up lactic acid? So they can regenerate NAD+, so that they can make more, they can go through glycolysis again. Because some energy is better than no energy. All right, who does alcoholic fermentation? Yes, which one in particular do you need to know? Yeast. You gotta know yeast. Yes. Number 10, what type of respiration are lactic acid and alcoholic fermentation? We only have two types of respiration. So what do lactic acid and alcoholic fermentation have in common? They're anaerobic. There's no oxygen. So the answer there is anaerobic respiration.
What's it says?
I don't know. We gotta do photosynthesis first. It'll be after photosynthesis. Number 11. What is the first step of all types of respiration? Glycolysis. Good. Number 12, how many ATP are created net from this first step of respiration?
2.
2. Number 13, where do the electrons go from the first step? Where would I find them? Right after glycolysis. They're in?
The link step?
Nope.
NADH.
NADH. All right. Number 14, where do all electrons end up at the end of aerobic respiration? So final destination molecule.
CO2.
No.
Oxygen.
Yeah, I would take oxygen or water. I won't give you both, okay? Generally, water is referred to as the final electron receptor. Of course, to receive it, it becomes water, so you'd be really, really wrong to give you both as an option. They're both good choices. Number 15, where do all electrons end up at the end of anaerobic respiration? You got two choices.
Carbon dioxide?
Nope. You're thinking though, keep going. Think about anaerobic respiration. Nope. You got to make more NAD+, which means you have to get rid of your electrons that are in NADH, which means you have to have somewhere to put them. It's the whole reason that when you run out of oxygen, you do this. What's the byproduct you make?
Glucose?
No?
Oxygen.
No? Anaerobic. So your muscles aren't getting enough oxygen to keep up with the demands that are put on them.
Lactic acid?
Lactic acid is one, and the other is?
Alcoholic?
Alcohol. Right? You only do lactic acid. Number 16, what is the potential ATP yield from fermentation? This is only the fermentation side, not the glycolysis side. So if we talk anaerobic respiration, there's two steps. You got glycolysis and you got fermentation. If we talk aerobic respiration, you got glycolysis and then you've got Krebs cycle and electron transport chain. So we're talking the anaerobic side. We go through glycolysis and then it has to go to fermentation. I want to know out of the fermentation, well, let's back up. How many are made again in glycolysis?
2.
2. How many are made in fermentation? 0. 0. Fermentation does not give you any energy. It simply gives you the ability to do glycolysis again because it stores your electrons.
So this is like 0 ATP?
Yeah. Yes, sir. All right, back side.
Wait, what's it?
Oh, sorry. What is the potential ATP yield from the Krebs cycle in ETC? It's 2 and 34, so it's 36. Okay. Number 18. Why would an organism ever use aerobic respiration? I typed those backwards. Those are exactly backwards. Why would an organism ever use anaerobic respiration rather than aerobic respiration?
Because a little energy is better than none.
This is true, because a little energy is better than none. What's the reason? What caused them to do it?
Because they don't have
Because
oxygen.
you ran out of oxygen. Yeah, very good.
So what would we put down for that? Just lack of oxygen?
Lack of oxygen, because a little energy is better than none. Those are both really good. Number 19, where does anaerobic respiration take place?
Mitochondria.
Anaerobic,
Oh,
cytoplasm. Think about it, bacteria can't do aerobic, so you can't say anything about a mitochondria. Number 20, where does aerobic respiration take place? Mitochondria. Good. Number 21, where would you find the electron transport chain?
mitochondria.
Yes, let's be more specific.
On the membrane.
On the which membrane?
Second.
Intermittent.
Inner membrane. Good. Questions like these, if you get that drawing down, it's going to help you. Okay. Where does the link step and the Krebs cycle take place?
Mitochondria.
It's in the mitochondria. Where?
Inside the membrane.
Inside which membrane?
Yeah.
Inside the inner membrane, which is called, that area inside the inner membrane is called the matrix.
So 22 is the matrix.
Yes. I wanted to make a Keanu Reeves Neo joke, but I think you guys are probably too young to know about that anyways. Have you guys seen The Matrix? You have no idea what I'm talking about, do you?
I heard it.
It was a phenomenon when it came out. You have no way of understanding. You should watch it. I highly commend it to you. In spite of the fact that I'm sure there's cussing and a lot of people die, it's still, the philosophy in it is just, wow. I would even let my children watch it one day. Not when they're 6. Too soon. Too soon. All right. Where are we at?
23.
23. Where does glycolysis take place?
Cytoplasm.
In the cytoplasm. Why do organisms do respiration?
Get energy.
To get energy. Good. (25) Which types of biomolecules can be used for respiration?
All of them?
All of them. Starting to get technical here. What is the name for the 3 carbon products of glycolysis?
Pyruvates.
Pyruvates. 27, what is the name of the 2 carbon products of the link step?
Acetyl-CoA.
Acetyl-CoA. I'm going to give you guys an option. If you want, I will double the value of this test to help you. That would, like, so just imagine if you got an A on this test, what that could do, to double the value of it, to make it worth twice as much as any other test. I'm not saying you have to do it. You can decide afterwards, but I'm just letting you know, like, throwing you a lifeline. I'm willing to double the value of this test to help pull us out. Some people are smiling, some people are like, I don't know, I don't trust him. All right. I would take that deal. I'd work hard for that deal. I would dare to say you'd be foolish to not take that deal.
But is the test going to include stuff from photosynthesis as well?
It will.
Will?
Yeah. Huh? You're gonna get, the test is gonna be what it is no matter what. I'm just trying to give you hope. I'm going, hey, you who are hopeless right now, let's do this.
Okay, all right, chill, not too much.
Hey, don't forget, like, I record these and I put them on Moodle. You could go back, you know, if you needed stuff, you could like pause it and answer the question, or, you know. Anyway, where are we at?
28.
28. What is the name of the 1 carbon products of both the link step and the Krebs cycle?
Carbon.
The 1 carbon molecule. Yes, it's carbon. What form is it in?
Monoxide?
Carbon dioxide.
Carbon dioxide. You said dioxide. I'm sorry, I didn't hear you. Carbon dioxide.
Oh, I spelled it totally wrong.
Did I spell it?
Oh, I could have spelled it wrong. Me.
Oh, yeah, I did.
No, you could have seen.
All right, chill. I didn't mean to. Chill.
Wait. What did you? That's what I was saying. I spelled it wrong.
All right.
Oh, the O.
I ain't mad at you. I can't speak.
What word?
Starting with the O.
What word?
Leave this poor girl alone.
No,
I'm
what?
gonna put a naughty finger up, and you're not
Bro,
gonna
what
like it.
word?
Put an O up. Here we go. Number 29.
Yes, I spelled it wrong too, bro.
Here we go. 29. Describe what happens in the electron transport chain. Now that got tougher, huh?
Dead silence.
That happens, right? So, if somebody came up and they're like, what did he draw? Can you explain this to me? What would you say happens?
Electrons flow through, right?
Good. Electrons are flowing through the electron transport chain. What happens as the electrons flow through?
They get diffused and turn into
More electrons, right?
My paper's not done.
Or what are the things on the top called? Like the,
They're H plus ions.
then don't they sometimes get turned into that?
Well, every time I bring in NADH and take the electrons, NAD plus goes out and the H's get left behind.
Oh,
They-
okay.
the pluses are the H's, okay? Because, take a look. A plus it's a proton, it's just a proton. H plus, that's just a proton. It's hydrogen that's been stripped of its electron, it's an ion, right? Has a charge, a positive charge, so I'm just drawing a plus. So whenever I bring in NADH, 2 electrons get stripped. One of the things that happens here is that this becomes this. The other thing is that NADH becomes NAD plus, and it's ready to go back and get more electrons. So NADH brings in 2 electrons, and I guess I could say a hydrogen ion also. And what's the first thing that happens? What happens to the electrons?
They flow out.
They flow down, right? It's a transport chain. So they're getting transported down a chain of molecules. The energy from those electrons is used by that chain to accomplish what? What do you see happening? You got arrows happening all over the place. What are they doing?
Moving.
They're moving what?
Energy? Electrons?
Hydrogen.
Every time that an electron flows through, this moves up. Right? These are pumps. They're literally pumping. What are they pumping?
Oxygen, protons?
Protons. They're pumping protons from here to here. That creates a concentration gradient. Okay? So ultimately, I end up with just a ton of these up here. You with me? Because I'm pumping. Every time 2 electrons flow through, I'm pumping hydrogen ions, pumping hydrogen ions, right? So I get a ton up here, I get very few down here. This is hypo, or isotonic up here.
Hypertonic.
Hypertonic. Take a look. Don't miss this.
Hyper.
Like I say, this is hypertonic. Tons of these. This is hypotonic. Everything wants to flow down its concentration gradient. I got a lot up here. I got a few down here. These want to move down. But I keep pumping them up. I'm using the energy from electrons to pump them against their concentration gradient to make more of a difference. They want to move back down. This is literally like a battery. In a battery, if you touch the 2 sides, like think of the big, like deep, whatever, what are those squares called? Are they D batteries? Here. If you touch the 2 sides of the battery, what happens to the electrons? They flow from one side to the other until it's done, right? The battery's dead because electrons have all flowed, and now they're equally distributed in the 2 cells, okay? These are like 2 cells, except instead of electrons, I've got protons. They want to flow from here to here. Once they're isotonic, they'll stop flowing, but I keep moving them up. So there is energy here. Just as much as there's energy stored in a battery, there's electrical energy stored here. They want to move here. This molecule, ATP synthase. This bad boy uses the energy of these flowing back to make ATP. It's literally a machine. Just as surely as you can plug a pencil sharpener in and put a pencil in and it'll sharpen pencils, this will allow hydronium ions, H+ ions to pass through, and for every 3 that pass through, it slams together and makes ATP. That makes sense? Do you know how amazing this is? Do you think that this just happened? Because evolutionists will tell you this just happened. Oh, there just happens to be this perfect little biological machine that makes ATP. Oh, there just happens to be this little factory inside of your cells that take all this stuff and take the electrons and run them through and pump things and create concentration gradients. That just happened. That did not just happen. You are fearfully and wonderfully made. This is one of the most amazing, I mean, it's microscopic, it's submicroscopic, and yet this is one of the most amazing things in all of creation. It is so incredible. Obviously, I did not do a good enough job talking about ATP synthase with you. Let me show you, I'm going to do an animation of just ATP synthase, okay?
Yeah, I looked at ATP for dummies and I watched a kid's video and it was very helpful.
All right, here we go.
We're making a big step.
Can I get, another set of everything you gave us to give to Allie?
Yes. Give me a second, okay?
I think she's sick.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
When will it stop working?
The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems,
If it ever stops
a
pumping
gradient
hydrogen
is
ions
always
up,
maintained. The
there won't be a
mitochondrial
concentration
hydrogen
gradient left anymore, and then it
is
won't
generated
turn.
as electrons.
You have to have a
in
difference, a
between
gradient, for that
processes.
thing to move.
That process can be seen in the mitochondrial electron transport chain animation.
What the fun video?
What?
There's like, I can't remember what it's called, now I gotta find it.
Well, not right now. Give me a minute.
Oh.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1,
You don't need to know these
cytochrome
names.
oxidase, and the complex that makes ATP.
You do need to know that.
ATP synthase. In addition to these complexes, 2 mobile carriers are also involved, ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation.
You want to see anything else before we go?
This animation illustrates 2 full cycles of electron donation.
You see anything on glycolysis or Krebs cycle or anything like that?
I have my good videos.
I hope they're good.
They're great.
And you got to keep in mind is these are the ones that I like. And I write the tests. I've been showing these videos for like 20 years. They're good. All right, anything else? Let's pray.
Wait. Are we not gonna finish our study yet?
Didn't we finish our study yet?
No.
Oh, well,
Not
let's
quite.
finish
We
our
have
study.
like 3 more questions.
Okay. Yeah, let's do that. What do we got?
We're on 30.
Number 30. What is the energy source that feeds the ETC? Electrons, yes. Good.
But what would you say 29 is?
All that. I'll put those videos up. Know them. The electron transport chain is incredibly important. There's one in a mitochondria. There's also one in a chloroplast. So this concept does not go away. We'll be dealing with it again during light reactions. Except it won't be electrons feeding it. It'll be light that's causing it. But very, very important. All right. So electrons feed the electron transport chain. Number 31, what is the energy source that is ultimately utilized by ATP synthase to make ATP? It's not electrons. What is it? What caused ATP synthase to turn?
Is it,
Protons. The concentration gradient. Either of those would be the idea, right? (31A), what creates this potential energy source? How did I get all the hydrogen ions up there? Kept moving them. It's drawn up there. Move something against its, no, that's going to be at the end. Move something against its concentration gradient, requires energy. What are they called? Made of protein.
The electron transport chain?
You got a bunch of water in your pool. and you want to get it out of your pool. It's not just going to go out. You got to use a pump. It's a pump. You have three things that are pumping hydrogen ions there. Okay. Number 32, draw, label, and describe the structure of a mitochondria.
Is that just the picture that you gave us?
Well, yeah, I don't know that I need as much detail as all that, but it'd be good. All right, what's this?
So,
Just, it's the membrane.
Mitochondrial membrane, what's this?
Membrane.
Inner membrane, what's this?
Matrix?
Intermembrane space. Inter means between the two membranes. This is the space between the two membranes. And this is called
matrix.
matrix. Watch the movie. All right, out here is
Cytoplasm.
cytoplasm. What happens in here? Two things. L and K happen in there. Link and Krebs happen there.
Wait,
Yes.
on the part that's in between the two membranes,
Yeah.
you call it the intermembrane,
Mm-hmm.
that's all.
Mm-hmm.
And also, the other one's called, like the actual membrane part is called the intermembrane?
Okay, this is the membrane.
Yeah.
This is the inner membrane.
Oh, yeah.
This is the intermembrane space. This is the matrix. Right? This is where the link and the Krebs cycle happen. This is where I'm trying to create this huge concentration gradient. This is where the ETC happens. On the inner membrane. You do not have to be able to draw all that. Okay? You just need to know that that is happening on the inner membrane. This is your intermembrane space. This is your matrix. Okay?